So Chapter 4 now continues to look at inheritance patterns, and we mostly look at the inheritance patterns in humans in this case. There are some genes in humans that do exhibit classical Mendelian patterns. For example, this is one where you can roll your tongue. You should try that. And this is hitchhiker's thumb. And these are single gene inheritance patterns here. We've already mentioned albinism as a recessively inherited trait. And we have already mentioned that you can't actually do crossing experiments on humans for ethical reasons, of course. You should by now know all the symbols used in pedigree construction, as we previously covered. And using these symbols, we can create a particular pedigree based on the information that we have. The goal, of course, is to utilize existing genetic data to create a map of inheritance, and so we might be able to predict who might be carrying what genes. So if you know your past history, genetically speaking, you can predict the future, genetically speaking. So the proband is, in this case, the number 2 here. This is the person with the arrow, and this is a diagram that we had before. And we've also seen this diagram before showing 4 generations of pedigree charts. But of course, patterns of inheritance can vary depending on the trait that we're looking at. For example, you can have autosomal dominant or recessive, X-linked dominant or recessive, even Y-linked or mitochondrial inheritance patterns, and we're going to look at each of these throughout this chapter. So let's start with some autosomal recessive traits. And here's albinism that we talked about up here. Here is cystic fibrosis, which we will talk about as well. Phenylketonuria, which we've talked about in the past. And sickle cell and xeroderma, which you may have heard about, where skin is very sensitive to UV light. And then there's a rather nasty disease called Tay-Sachs disease as well. These numbers over here, these OMIM numbers, OMIM stands for the Online Mendelian Inheritance in Man, and what that is an online database of all currently known human traits. And so here's a screenshot of the web page for OMIM, and if you just search for OMIM, and you will come up with this website. Very, very useful resource. So let's look at this autosomal recessive trait of cystic fibrosis. Cystic fibrosis controls the movement of chloride ions across a plasma membrane. Here's a diagram of a plasma membrane here. Here's the phospholipids. Right in there. Here's the bilayer. Here's the other side of the plasma membrane. Here are the phospholipid chains. And across those bilayer membranes are some proteins that allow chloride ions into and out of the cell. Well, they regulate the transport of chloride ions throughout the cell. This gene is on chromosome 7. Is at a location called Q31.2. Now what this means is that we give the letter P for the short arm of a particular chromosome. A particular chromosome, any particular chromosome, has a centromere, right in here. And one half of that chromosome is going to be shorter than the other half of the chromosome. So the short arm, they give the term P, and the long arm, they give the term Q. So if you have a gene at Q31.2, it means the long arm, and it means 31.2 units from the centromere. So that's the genetic component of cystic fibrosis. The biochemical component is that when a mutation occurs in the cystic fibrosis gene, it produces a mutation in this particular protein right here, and that affects these transmembrane proteins here. The result is an osmotic imbalance occurs in between inside and out of the cell, and there are many phenotypes resulting from this, but probably the most commonly known is a difficulty in breathing and salty tasting skin. Those are 2 diagnostic tests for cystic fibrosis. Another recessive gene trait is the infamous sickle cell anemia. This is the production of mutant hemoglobin, which changes the shape of cells, decreasing their ability to carry oxygen. It's also autosomal, which means it's on chromosomes 1 through 22. And this is sickle cell over here, and this is normal. These are normal cells over here. You can see the biconcave component of a normal red blood cell. This is because the hemoglobin molecule is a standard globular shape. Over here on the right, the sickle cells are caused by sickle-shaped hemoglobin. So the hemoglobin molecule is a different shape, and it sort of looks like a sickle. It's a very long shape, and so it points, makes these red blood cells very pointy, as you can see here. Here is a pedigree chart, and here is a son with that particular trait. We can tell they're the proband because they have this arrow. Remember, the proband is the first person to come and see about the particular trait. And if this is recessive, then we know that this person must be little a, little a. That must be their genotype. Well, we know they got one little a from one parent, the other little a from the other parent, so that the genotypes of these parents must be something little a, each one. But because each of these parents do not have that trait, then we know that these parents must be heterozygous, because they do not have the trait, but they passed on the recessive trait to each of their parents, to this particular child here. This is shown in a Punnett square. We can see it's a standard heterozygous monohybrid cross, and we see from two heterozygous parents we can have one homozygous dominant, two heterozygotes, and one recessive down here. So that recessive, of course, happened to child number 2. We don't know what these numbers 2 and 3 are. They could in fact be homozygous dominant, or they could be heterozygous. We don't know. Here are some autosomal dominant traits. These are traits, obviously, that you only need one dominant allele to get that trait. So you can be big A anything, and that you will have the trait. There's achondroplasia, which is dwarfism, which means if you are not have achondroplasia, then you are double recessive. There's a few others. Hypercholesterolemia, which is high levels of cholesterol. That's also another dominant trait. Huntington's disease, which is a nasty one. Marfan disease, which we will talk about in a minute. And then porphyria, which if you've ever seen the movie The Madness of King George, porphyria is one of the disease that they are talking about in that movie. So autosomal dominant traits, every affected individual has at least one affected parent, and two affected individuals can have an unaffected child. So how does this work? Well, if you have here is a father and here is a mother. If the father has the trait, they are big A something. If the mother does not have the trait, they're double recessive. If you pass that on to, say, a couple of children, here's a daughter and here's a son, then we know that the mother can only pass on one recessive trait, so that's going to be here and here. And the father is either going to pass on this dominant trait or whatever's here. Now, if we make him heterozygous, then let's say this recessive A is passed on to this second child, and this dominant A is passed on to the first child, just purely randomly, of course, then we show that this child here also has the trait. So every affected individual, for instance, child down here, has at least one affected parent up here. Now, two affected individuals can have an unaffected child. So here's two affected individuals. Here's mother and father. Both have the trait. And here are a couple of children that do not have the trait. So how does this work? Well, just a standard Punnett square. If both parents are heterozygous, then here is the unaffected child. Remember, these are dominant traits. And so here's heterozygous mother and father, and here are a couple of unaffected children. Here's a large pedigree chart for a dominant trait, and remember the rules from before. Every affected individual has at least one affected parent, and two affected individuals can have an unaffected child. So let's see how we're looking over here. Here is an affected person, so they must be, say, big A something, and here is their mother, and their mother must be big A something. Father is double recessive in this case, so this child must be heterozygous. Remember, it's an autosomal dominant trait, which means you've got to be big A something to have that trait. We had mentioned earlier something called Marfan syndrome. This is an example of an autosomal dominant genetic disorder, and it's reasonably famous due to its sports connections. For example, somebody can be quite tall, which of course usually gets them into the ball throwing sports, but one of the problems with Marfan syndrome is that you have a very weak aorta, which of course can be a problem. Here's what happens. Here's the aorta, which is the main blood vessel coming out of the heart, and it is affected in Marfan syndrome. It becomes weak, and what will happen is that under extreme stress, this aorta will rupture, which of course leads to instant death. Here's another mechanism of inheritance, and it's called sex-linked inheritance. And this is where genes are on the sex chromosomes. They're not autosomal genes, they're not autosomal chromosomes, they are sex chromosomes. And the sex chromosomes are, of course, in humans, X and Y. And males pass on an X chromosome to all of their daughters, and females pass on an X chromosome to all of their children. How does this work? Let's do one easy Punnett square. If we have the male here and the female here, we're only concerned with sex chromosomes, that's the X's and Y's. Here is the mother. The mother passes on an X chromosome to all of her children. The father passes on an X to his daughters and a Y to his sons. So let's look now at some of the genes on these sex chromosomes. When we say there's a gene on a sex chromosome, we say it's linked to that particular chromosome because it's physically connected, because it's part of the same molecule. And so we have the term X-linked for genes that are on the X chromosome, and not surprisingly the term Y-linked if the genes are on the Y chromosome. So we're going to look at the pattern of inheritance on both of these particular cases. Now, there's one new term here, it's called hemizygous. And if you look at males, they have one X and one Y, so you only have one of the X chromosomes. And so it's called hemizygous for X. But of course you only have one Y chromosome as well. And so it's also hemizygous for Y. So hemizygous just means it's just one gene present, not heterozygous and not homozygous, but hemizygous. So here's a standard sex chromosome Punnett square. Here's the male, X and Y. Here's the female, XX. And as we already drew out a little earlier, here's the female offsprings, and here's the male offspring on the right-hand side. You might notice half the progeny are going to be female, half the progeny are going to be male. That's why there is roughly 50% males and 50% females in the world. So before we leap into what these sex chromosomes do, there's a little bit of history. Where did the discovery of sex chromosomes come from? So let's go all the way back to 1910. There's a gentleman called Thomas Hunt Morgan, and Thomas Morgan, he ran a fly lab, commonly known as the fly lab. He looked at Drosophila flies, otherwise known as fruit flies, and he was doing genetic experiments with fruit flies, and you can see here it basically used all old milk bottles with some crushed up bananas in the bottom and some cotton wool on the top, and had thousands and thousands of flies doing experiments on them.